For most of the 20th century, geneticists searching for the deepest roots of the entire human species looked in the wrong place, assuming the oldest, most divergent lineages of modern humanity would be found somewhere near where the first fossil evidence of Homo sapiens had already been discovered, in eastern Africa. Then researchers began sequencing the genomes of a small group of indigenous hunter-gatherer and herder communities living in the deserts and coastal regions of southern Africa. And the results didn't just add a new chapter to the human story. They pushed the entire timeline of when our species actually began to diverge from itself back by more than 100,000 years, and revealed that the single deepest split in the entire family tree of every human being alive today runs directly through a population most of the world had never heard of. The population at the center of this discovery is collectively known as the Koisan, a term used by researchers to describe a genuinely diverse group of indigenous communities native to southern Africa, encompassing both San hunter-gatherer groups and Kukui pastoralist or herding communities, historically referred to in older literature by terms including Koisan, San, or Bushman, though many of these older labels are now considered outdated or, in some cases, genuinely offensive by the communities themselves, with Koisan or Sankuko generally preferred in current scientific and cultural usage. What unites these communities beyond their shared geographic homeland across parts of modern South Africa, Namibia, Botswana, and neighboring regions is a shared linguistic heritage built around click consonants, a genuinely distinctive feature of the Koisan language family found almost nowhere else on Earth in comparable form, and, as genetic research would eventually reveal, a genuinely extraordinary shared ancestral history reaching back further into the human past than that of any other population scientists have yet studied. The first serious genetic investigation into just how ancient this population's roots actually ran began building momentum in the early 2000s, but the true scale of the discovery only became clear with a landmark study published in the journal Science in September 2012, led by researcher Carina Schlebusch of the University of the Witwatersrand and Uppsala University, working alongside senior collaborators including Himla Sudial of the University of the Witwatersrand and Matthias Jakobsson of Uppsala University. The research team analyzed genetic data from 220 individuals sampled across 7 different Koisan groups spanning a wide geographic area of southern Africa, examining approximately 2-3 million distinct genetic variants per individual, at the time the largest genomic study of its kind ever conducted specifically on southern African populations. The result, the study's authors themselves highlighted as most significant, was direct and, for the field of human population genetics, genuinely paradigm-shifting. The deepest genetic divergence found among all living human populations, anywhere on Earth, occurred within this specific southern African lineage, splitting away from the ancestral line that would eventually give rise to every other population on the planet somewhere around 100,000 years ago, well before modern humans ever began migrating out of Africa, and roughly twice as old as the next deepest population divergences researchers had previously documented among Central African pygmy populations and East African hunter-gatherer groups. As Jakobsson put it directly in comments accompanying the study's publication, these deep divergences carry real significant implications for how researchers understand the entire history of humankind, since the pattern of genetic variation the team uncovered suggests human populations have been genuinely structured, meaning meaningfully separated from one another, for a considerably longer stretch of time than earlier models had assumed, raising the genuine possibility that modern humans as a species emerged from an ancestral population that was never fully homogeneous or unified to begin with, even at its earliest stages. This finding effectively established the Koisan as representing one branch of the single earliest population split within the entire species Homo sapiens, with every other population on Earth, every European, every East Asian, every other African population, and every indigenous population in the Americas or Oceania, descending from the other branch of that same original ancient divergence. That same 2012 study revealed something else genuinely significant beyond simply establishing the age of the divergence itself. By modeling how the effective population size of the ancestral Koisan lineage fluctuated across tens of thousands of years, researchers found clear evidence that this population's numbers rose and fell in ways that closely tracked major climate shifts documented independently in the paleoclimate record, expanding during wetter, more hospitable periods and contracting sharply during arid phases when the southern African interior became considerably less habitable. This finding mattered because it demonstrated for the first time with this level of genetic precision that climate itself functioned as a direct, measurable force shaping the population history of the deepest surviving human lineage on the planet, tying the genetic record directly to the physical, environmental record in a way that considerably strengthened confidence in the overall dating and interpretation of the study's core findings. That 2012 study, remarkable as it was, turned out to represent only the opening chapter of a considerably longer scientific investigation because sequencing modern DNA alone could only push the story so far back with real precision. To go deeper, researchers needed something considerably harder to obtain: actual ancient DNA recovered directly from human remains that had lived and died in southern Africa long before recorded history began, providing a genuine physical snapshot of the region's population at a specific datable moment in the deep past, rather than a reconstruction built entirely from working backward through the DNA of people alive today. That opportunity arrived in 2017 when a research team once again, led by Karina Schlebusch, this time in expanded collaboration with an international group of researchers, successfully sequenced the genomes of 7 ancient individuals recovered from archaeological sites in KwaZulu-Natal, South Africa. Published in the journal Science under the title Southern African Ancient Genomes Estimate Modern Human Divergence to 350,000 to 260,000 years ago. The remains analyzed included individuals from sites called Ballito Bay and Duneside, dated to the Stone Age and representing genuinely ancient hunter-gatherer populations who lived in the region before any later migration from within or beyond Africa had reached that specific area, alongside additional Iron Age individuals from nearby sites including Newcastle, Eland Cave, and Mfongosi, providing researchers with a genuinely rare, direct comparison between the region's deep ancestral population and later arrivals who eventually settled in the same general area. The genetic results recovered from these ancient remains were nothing short of extraordinary in their implications. The 3 oldest Stone Age individuals from Ballito Bay and Duneside carried mitochondrial DNA belonging to haplogroup L0D, a lineage still common specifically among modern-day Koisan populations today, providing direct physical, genetically verified continuity connecting this deep ancient population straight through to the people living in the same region right now. Both of the male individuals recovered from Ballito Bay additionally carried a specific Y chromosome haplotype, A1B1AB2, which likewise remains common among modern Koisan communities today. This represents about as close to direct, unambiguous genetic proof of population continuity as ancient DNA research is capable of providing a clear, physically documented paternal and maternal lineage connecting people living in southern Africa today directly back to hunter-gatherers who lived in the exact same region long before recorded history. By contrast, the more recent Iron Age individuals recovered from the same broader region carried an entirely different mitochondrial signature, haplogroup L3E, a lineage common today, specifically among Bantu-speaking populations, providing direct physical genetic evidence of the later, well-documented Bantu expansion that gradually spread agricultural and pastoral populations across much of southern Africa in the centuries and millennia that followed. Using this direct ancient genetic data, combined with sophisticated computational modeling of mutation rates and population divergence patterns, the research team arrived at a genuinely striking revised estimate for when the ancestors of modern humans first began to genetically diverge from one another as a species. Rather than the roughly 100,000 years the 2012 study had already established using modern DNA alone, this new ancient DNA evidence pushed that estimate back considerably further to somewhere between 350,000 and 260,000 years ago. This finding carries genuinely profound implications for the broader question of where and how Homo sapiens first emerged as a distinct species, because it places the very earliest population divergence within our species at a time, depth roughly comparable to, or even predating, the oldest currently known Homo sapiens fossil remains, and it directly reinforces a growing body of separate archaeological evidence pointing towards southern Africa as one of the genuinely significant regions where our species may have first taken shape, rather than supporting the older, simpler model that treated eastern Africa as the sole point of origin for all of modern humanity. The archaeological record independently supports exactly this kind of deep, continuous human presence in this specific region. Middle Stone Age sites in KwaZulu-Natal, including Sibiru Cave, Umlatuzana Rock Shelter, and Border Cave, demonstrate confirmed continuous human occupation stretching back more than 100,000 years, providing an independent, physically excavated archaeological timeline that lines up closely with what the genetic evidence separately and independently indicates about the deep antiquity of human settlement in this specific corner of the African continent. This convergence between two entirely separate forms of evidence, genetic modeling on one side, physical stone tool, and habitation layer excavation on the other, is precisely the kind of independent corroboration that gives researchers real confidence in a finding this significant, since neither line of evidence depended on or was influenced by the other when each was originally developed. Beyond simply establishing the extraordinary age of this lineage, subsequent genetic research has revealed that the broader Koisan population itself is considerably more internally diverse and genetically complex than a single uniform label might suggest. A further landmark study, published in 2020 in the journal Molecular Biology and Evolution, and again led by Schlabusch's research team, sequenced 25 complete, high-coverage genomes drawn from 5 distinct Koisan groups, deliberately chosen to represent all 3 of the major linguistic branches within the broader Koisan language family spread across an extensive geographic range of southern Africa. This research confirmed that the Koisan population as a whole carries the single greatest level of genetic diversity documented anywhere among modern human populations globally, with fully 25% of the genetic variants identified in this specific study found to be entirely unique to Koisan populations, not present in any other human population sampled anywhere else in the world. An earlier complementary study, published in the journal Nature in 2010 and led by researcher Stephen Schuster, included the full genome sequencing of an indigenous hunter-gatherer from Namibia's Kalahari Desert, alongside additional partial genome sequencing from several other Khoe-San individuals across different regions of the Kalahari, and notably, the full genome of Archbishop Desmond Tutu, included specifically as a comparison point representing a Bantu-speaking South African individual. That 2010 study made a genuinely remarkable, almost counterintuitive observation directly in its published findings. The specific Koisan men sampled in that study showed greater genetic differences from each other, individually, than a typical European individual would show compared to a typical East Asian individual. a finding that powerfully illustrates just how much raw genetic diversity remains concentrated within this single population, compared to the comparatively narrow genetic variation found across the rest of the entire human species combined. The same study identified more than 1 million previously undocumented genetic variants across the sampled genomes, including thousands of specific amino acid variants never before recorded in any human genetic database, underscoring just how significantly underrepresented Southern African populations had been within global genomic research up to that point. This extraordinary internal diversity connects directly to a genuinely important distinction within the broader Koisan population itself, one that later genetic research helped clarify considerably. The term Koisan actually encompasses two historically and culturally distinct groups: San hunter-gatherer communities, who maintained a foraging lifestyle across many thousands of years, and Cocos pastoralist communities, herders, who adopted livestock husbandry considerably more recently in the region's history. Genetic research examining this specific distinction found that co-pastoralist populations descend from an earlier southern San hunter-gatherer group that appears to have adopted pastoralism following genuine genetic contact and admixture with an East African population, one that most likely served as the original source, bringing pastoralist practices into southern Africa for the first time. Researchers identified this connection partly through specific lactase persistence genetic variants, the same broad category of gene responsible for adult milk digestion ability studied extensively in other populations worldwide, found present in co-pastoralist populations at frequencies indicating a real, documented East African genetic contribution layered on top of their older, deeper San ancestral foundation. A genuinely elegant example of how a specific cultural and economic transition, the adoption of herding as a way of life, left behind a directly traceable genetic fingerprint that researchers can still detect and interpret today. The broader genetic history of southern Africa also reveals a genuinely fascinating pattern of population contact and absorption, extending well beyond the Koisan population's own internal history, connecting this ancient lineage to the considerably more recent and geographically expansive Bantu expansion that reshaped population patterns across much of sub-Saharan Africa over the past 2,000 to 3,000 years. Research examining a specific mitochondrial lineage called Khoe-San, itself a deeply divergent branch closely associated with Koisan-speaking populations, identified three distinct sub-branches: Khoe-1a, Khoe-1b, and Khoe-2. Strikingly, only the Khoe-1a branch remains present today among actual Koisan-speaking populations living in Namibia and Botswana, while the other two branches, Khoe-1b and Khoe-2, are now found almost exclusively within Bantu-speaking populations settled specifically in Zambia. This distinctive geographically split pattern provides genuine, direct genetic evidence of a specific historical process. Ancient Koisan-related populations that once inhabited considerably wider stretches of eastern and central Africa were gradually absorbed into the expanding, numerically much larger Bantu-speaking population. As that expansion swept across the region, their distinct maternal genetic lineages surviving and remaining detectable today, but now carried forward almost entirely within communities that no longer speak a Koisan language or maintain a distinct Koisan cultural identity, a genuinely poignant genetic record of cultural assimilation occurring alongside real, measurable biological continuity. This same broader pattern shows up again in separate research examining Koisan-related genetic ancestry detected specifically in modern Eastern African populations, geographically distant from the core Koisan homeland in the far south of the continent. Researchers found that this Eastern African Koisan-related genetic signal is related in roughly equal measure to the deeply divergent lineages identified in the various different modern Koisan groups studied further south, indicating that the specific Koisan-related populations who once settled in eastern Africa were already genetically distinct from the populations remaining in southern Africa by the time this particular divergence occurred. A further layer of genuine complexity within what might otherwise be mistakenly assumed to be one simple, unified ancestral population. Ongoing research into this population has continued directly through the current period, with a further study published in the journal Nature Communications specifically dedicated to cataloging the genetic variation found across the earliest diverging populations still living today, once again confirming and refining the picture of the Koisan as comprising several genuinely distinct populations rather than one single internally uniform group, even while sharing this same remarkably deep common ancestral root. Beyond establishing the extraordinary age and internal complexity of this lineage, researchers studying the Koisan genome have also identified real, measurable evidence of local evolutionary adaptation, genetic changes shaped specifically by the demanding environmental conditions this population has survived across many tens of thousands of years of continuous habitation in southern Africa. Specific gene regions connected to immune system function, muscle performance, dietary metabolism, and protection against ultraviolet light exposure all show distinctive genetic signatures in Koisan populations, patterns researchers interpret as evidence of natural selection acting both very early in the broader modern human lineage and, in some cases, considerably more recently, reflecting specific localized adaptation to the particular environmental challenges these communities encountered across their exceptionally long continuous history in this specific region of the continent. This growing body of genetic research has also carried real, direct consequences for how the scientific community itself engages with the Koisan communities, whose DNA has made all of these discoveries possible in the first place. Because earlier decades of genetic research into indigenous populations worldwide, including southern Africa, were frequently conducted without meaningful community consent, benefit sharing, or ongoing engagement, San community representatives worked directly with researchers to develop what became known as the San Code of Research Ethics, formally adopted in 2017 and recognized as the first research ethics code of its kind, developed and authored by an indigenous community anywhere on the African continent. The code explicitly requires that researchers seeking to study San genetic material engage in transparent, respectful consultation with community structures before any research begins, share findings directly with the communities involved rather than treating them purely as a source of samples, and ensure some measure of genuine benefit flows back to the communities themselves, a framework that has since influenced how genetic researchers elsewhere in the world approach research involving other indigenous populations, and stands as a genuinely significant, community-driven contribution to the broader ethics of population genetics research as a field. Put all of this accumulated research together, spanning more than a decade of increasingly sophisticated genetic investigation, from the initial 2012 discovery of the deepest population divergence in modern humans through the direct 2017 ancient DNA confirmation pushing that same divergence back to as much as 350,000 years, to the detailed 2020 mapping of the population's remarkable internal diversity, and a genuinely coherent, scientifically rigorous picture finally comes into focus. The Koisan peoples of southern Africa represent, according to the best currently available genetic evidence, the single deepest, most anciently diverged branch of the entire modern human family tree, a lineage that had already begun separating from the ancestors of every other person alive today at a time depth potentially reaching back beyond 350,000 years, likely predating, or at minimum closely coinciding with, the very earliest fossil evidence of Homo sapiens as a distinct species. That deep ancestral thread survived remarkably through ancient hunter-gatherer communities whose direct genetic descendants can be traced with real, documented precision straight through to Koisan communities living in the exact same region of southern Africa today, even as later waves of Bantu-speaking agricultural and pastoral populations swept across much of the wider region over the following millennia, absorbing considerable numbers of Koisan-related communities along the way while leaving other Koisan populations in certain specific regions genuinely continuous and culturally distinct right up to the present day. This is not a story about a population frozen unchanged in time. It's a story about a lineage that has spent longer than any other population on Earth continuously adapting, diversifying, and surviving, carrying within its own DNA the single deepest, most direct physical record of how our entire species actually began.